So good morning, everyone. My name is Lan, and now I am working with International University, the same university with Dr. Hạnh. And then today, I'm so happy to be here to present our study on feasibility of using Physarum polycephalum microplasmodial biomass as a feed for Artemia Enrichment. And then during my presentation, okay, I divide into like the four main parts. And then for the first one, related to the species we use in our study, that is Artemia. There are some reasons why we choose Artemia for our study. Firstly, Artemia, that is considered like the most widely utilized live food item in aquaculture, especially for laviculture, including shrimp and fish larvae. And secondly, Artemia is they are the filter feeding organism, and so they are considered as a perfect carrier to carry essential nutrition, and then other kind of element we would like to transfer to shrimp or fish. And so that is the two main reason is why we choose Artemia for our study. And so in the photo I bring here, you can see that there are so many different kind of elements. We can like use Artemia as a carrier to bring to fish or shrimp to increase the quality of fish or shrimp larvae. And then now is the most common product they are using to enrich Artemia, that is a DNA cell coat. For DNA cell coat, they consider it is like a polyunsaturated lipid, but they are so costly and easily to decrease the quality of water. And then in aquaculture, quality of water is a place of essential roles in is like maintaining is a good aquaculture system. Along with DNA cell coat, they also can use LC as another kind of products to increase the quality of Artemia, considered as a live food for as a different kind of aquaculture species. But for LC, they have a like the richest cells, make them is more difficult to digest. And so consider to Physarum polycephalum or plasmodial slammer, they have so many interesting features, such as is no cell walls, make this species is more digestible, more easier to digestible compared to like the LC. And then for the plasmodial slammer can grow quickly in the simple culturing condition, and also like the high quality nutrition in terms of nutrition. For example, here, like we can find out for plasmodial slammer, it contains about more than 11% of protein, and then in this like the lipid content, we can find out is so many types of unsaturated fatty acids, such as linoleic acid or oleic acid. So make them is like quite potential in applying it kind of food for the Artemia species. And so in our study, we design in 5 different treatments. So Artemia after 24 hours of incubating, so the nucleus of Artemia, we will use like we'll go through the enrichment process with 5 different diet treatment, including is only DNA cell coat. Okay, like I mentioned before, that is like unsaturated lipid sources. And then for other 2, it is a combination between is a DNA cell coat, which is a dry form or is a fresh form of slammer. And then for other 2 treatment, that is a contain only dry microplasmodial biomass or the fresh microplasmodial biomass. Okay, after 6 hours of enrichment process, okay, we will collect Artemia napoli to determine the length to identify growth performance of Artemias. We also like collect Artemia to analyze the nutritional values, including is a total lipid, total protein, and total carotenoids. So firstly, mention about growth performance. If you look at the chart, we have right here like the four dry form Physarum, polycephalum, microplasmodial biomass is given the highest body length. And then for the lowest one, we have like a fresh form and also the early cycle in the treatment of early cycle. So for the combination, they have no difference compared to dry form of SLAMO. So mention about the protein content and even is for carotenoid contents. The result, the obtained data is so that for the cell core, they have a low it in terms of protein content. And then for dry treatment, is still have a like a high git protein contents. Sorry, for this one is like a combination of cell core and then fresh form have a high git protein content, and then the lowest one it is belong to the cell core treatment. It have a like the same result compared to carotenoid content. For carotenoid content, the combination between is a cell core and then fresh form bring is like a high results. And then the lowest one is we can see in the dry treatment. So for the lipid content, the cell core is have a high lipid content, but this result is no different when we compare between the combination of cell core and then dry and fresh form of SLAMO. And so from is our result. So I give the summarize what we found out after carry out our experiment. Okay, which is like a dry microplasmodial of SLAMO. So that is the best performance. Mean that is I have a like a highest data, and then the combination between is a cell core and then fresh form of is a microplasmodial. SLAMO obtain is both protein and then carotenoid contents, the highest protein and carotenoid contents. In terms of like the lipid content, okay, the cell core even it have a like a highest, but there are no significant difference between is a cell core and then the combination between cell core and dry and then fresh form. And so we can see that the combination of DNA cell core and then fresh form consistent through is like a protein, carotenoid, and even lipid. And so we conclude that microplasmodial biomass of SLAMO can be used as enrichment for Artemia. But like in our result, you can see that just the only use like the biomass, it is have a less effective than is a combination between is a cell core and then SLAMO. And so we would like to carry out some more study to find out to optimize the supplement procedure when we use SLAMO as is to feed Artemia and even is for other species. And then we also like a good lab to conduct to analyze the fatty acid composition because well, we did it before, but the result is not reliable. And so also like we would love to carry out more study after like enrich Artemia, which is a SLAMO. We can like use Artemia to feed on shrimp or fish, okay, to see more as effective of is using SLAMO as is like a kind of feed for apply in is a nutrition and also like the alternative feeding for so many kind of species. Okay, that brings me to the end of my presentation. It is quite short, but I would love to get any comments or even question from you.